Hello, my name is Scott McElveen. I'm here to give you a quick demo of BeCrop technology for crop advisors and farmers. BioMakers has produced a test that assesses the blind spot in our soil testing. As you know, soil has three main components, three legs to the stool: the chemical, the physical, and the biological. Our current testing overwhelmingly deals with the chemical and the physical. Not for any ill will, just there have not been high resolution, affordable, and agronomically actionable tests in the past in order to well assess the biological. What we do is we extract DNA from soil. We snip out what's called a marker gene. It's essentially like a fingerprint we can use to identify the organism that bears this genetic sequence. We identify that organism, and then what is unique about BioMaker's test is the database, the world-leading database of agricultural microbe taxonomic references that we've built over the course of the last decade that enable us to take these identifications and then class them into functional groups, into subgroups in the soil that perform a plant growth promoting or plant growth inhibiting process. We have our auxin producers, our nitrogen mineralizers, our nitrogen immobilizers, our suppressors of fungal pathogens, biocontrol agents, so on and so forth. Ultimately, we are able to provide indications about four main categories: overall microbiome health and functionality, plant growth promoting and growth mitigating microbes, everything from pathogens to pathogen controllers, plant growth hormone producers, and organisms that help plants adapt to stress. And then our nutrient cyclers. We focus on organisms that transform nutrients from our soil fertility test into a crop available form, and also those that eat from the same plate as the crop, so to speak, by utilizing the same soluble plant available forms that roots take up. We also look at carbon pathways and the capacity of our soil to mobilize 8 micronutrients. For the longest time, we've presented that as reports like the following, which are incredibly useful for diagnosing specific questions, for testing an underperforming area to a strongly performing area. You can also use our free tools on the BeCrop portal to compare between two samples or clusters of samples to identify likely candidates for the biological drivers that are producing a difference between two pieces of land which are not responding to the management tools that we're bringing to bear thus far. There are a number of excellent tools for comparing between samples and digging deeper into the data. The question is, how can we characterize a landscape? This was the challenge that we were facing as growers wanted to better understand the spatial variability in their microbiome, wanted to have a more affordable price to assess a larger portion of their land, and to leverage the data to provide recommendations. And so what we have done is we've utilized our tens of thousands of sample database of geolocated dated samples to better assess the relationships between crop behavior, crop responses as assessed through satellite imagery, and topography, abiotic environmental factors, and the DNA results we'd had off each of those samples. With that, we have now been able to take in shape files from someone who wants to test their fields. That land is analyzed on the basis of satellite imagery and the environmental parameters that I just referred to. And then it is zoned on the basis of the parameters that we know are drivers of microbiome composition and function. So that's a machine learning clustering algorithm, predicts biological zones, and then that system recommends sampling coordinates. And we have found that we can, for $5 an acre, $7 an acre, assess microbiological function across a landscape to a 10 meter by 10 meter resolution that is agronomically actionable and produces results in taking action on the recommendations that come out of the microbiome assessment and the characterization of your fields in relationship to our 400 plus field trial and growing database of biological and biostimulant trials. So let's take a quick look at a map. Here is a popcorn farm in Nebraska. We're looking at the capacity of the soil. Let's break it down a little bit. the capacity of the soil to mineralize nitrogen, to take unavailable organic nitrogen. Beg your pardon unavailable inorganic nitrogen, and release it into a soluble crop available form, ammonium nitrate. These yellows look similar, but this field on the right scores on average an 11 out of 100 in terms of our normalized scoring. These are in the 30s. Now let's look at the tendency of these soils to immobilize nitrogen, the competition for the exact same supply that our crop uses. That same field that had the lowest capacity to mineralize, to supply nitrogen in our system into a crop available form, also has the highest tendency to immobilize, compete for that same supply. And so when we take those two metrics in tandem, we call that global nitrogen, the tendency of our soil either to supply crop available nitrogen or compete for it. This field is the most likely to face a bottleneck. They all are somewhat poorly off, but this one's the most likely to face a bottleneck, and therefore is also the field most likely to respond to us taking measures to enhance crop availability, be it Y dropping, banding, be it supplemental feeding of foliar nitrogen, or the inoculation of a mineralizer and uptake efficiency enhancer such as a fulvic acid. If we look at phosphorus, we also see stark differences between these fields, particularly the north and the southeast, in the capacity of our soil to take unavailable inorganic phosphorus and turn it into crop available phosphate. Fortunately, if we do have any degree of organic matter on this field, residue, if there have been manure applications, then our capacity to mineralize unavailable organic phosphorus is rather good on this farm. So depending on what sources we know we have from our Mehlich 3, our Haney analysis, or what have you, then we may be in good standing with our phosphorus. But you can imagine scenarios where you'd be at risk not knowing this information, depending on what your phosphorus sources are and what your competition from the soil for soluble phosphate is. So in the case here, there's not heavy competition for soluble crop available phosphate. That's just looking at the nutrient cycles. As I mentioned before, we measure a great many other things, such as at-risk pathogens. These are field level risk scores in this earlier iteration of BeCrop Farm. In an upcoming update, just in the next month or so, these are going to become more high resolution metrics. But at the moment, for instance, if I look at Gauss's wilt, I can see that there are particular fields that are much higher risk. What's my capacity to deal with a bacterial threat? It's extremely low. It's undetected on this field, low on this field, a score of 1 out of 100, nearly the lowest score we've ever recorded in corn. There are, of course, our plant growth hormone producers, our stress adaptation microbes, our overall soil functionality, biodiversity, and sustainability scores. There's a great deal here, as well as the integration of our field trial data to recommend active ingredients that are most likely to address the needs of these fields, not just on the basis of the needs of the fields, but on the basis of an environmental match and a microbiome match between these fields and trial locations where those experiments were carried out. So if I jump right back to here, we have our diagnostic map. This is where you would see our needs-based recommendations for nutritional needs, disease risks, management practices improvement in terms of the sustainability score. There are PDF reports available as well. These are excerpts, one page out of several, for the field level reports and farm level reports. And it's also worth noting, of course, that our metrics do correlate with later in the season agronomic outcomes. Here we have nitrogen cycle health index taken in April. By July, it's not a perfect echo. Agroecosystems are complex systems, but there's clearly a mirroring of the health of the nitrogen cycle and biomass accumulation later in the season. I have lots more information that I'm able to share with you. Hopefully this is a teaser that helps you figure out whether you are interested or not, could make use of these sorts of novel insights or not. And I very much look forward to hearing from you. Thank you. Bye.